We use animal models in our research because we can experiment with new treatments in animals before these treatments are really ready for the clinic. And we're able to then really get answers that we hope will allow us to take the information back to the clinic and come up with new and even better treatments.
We can't say whether something new is safe or whether it works unless it's gone through a proper set of tests. These tests actually have to begin at the level of animals, so it's very important to do that.
We're actually reproducing the cause of the disease in the animal model, and that is a tremendous advantage.
Fruit fly models, worm models, mouse models, rat models, you can tweak the system with genetics that you can't do in other systems. The reason we study these living systems is because they have the appropriate structure so that there's cells that talk to each other. There's different cell types within a given region. When you have it in a dish, it's much more difficult. You can look at a single cell type, but not necessarily the complexity that happens in the brain.
With an animal model, we can look for visible changes, microscopic changes, biochemical changes within the mice that would be impossible to do in a living human being, for instance, and would also be very difficult to do, say, in a tissue culture dish.
Many people who don't understand animal research will say, well, why don't you use computer models? Why don't you use cells growing in a dish? The problem with computer models is they have to model something. You have to understand the process before you can actually use a computer to further understand it.
We've made a great deal of progress in developing animal models for Alzheimer's disease, such that we can now take a human gene from families who have what's called familial Alzheimer's disease, where this amyloid protein is deposited in the brain. We can take the gene out of people, put it into a mouse, and those mice will develop that same protein in the brain as if the mice were developing Alzheimer's disease. We can develop various therapeutics, drugs, to try to remove that protein from the brain, or perhaps we can block it being deposited in the brain. All kinds of combination therapies can be tested in animals relatively quickly, and then we translate that into humans.
Think about this in terms of if this was your child, and what would you really need to see in terms of evidence before you're ready to try this experimental treatment in your child?
If we can demonstrate that the human nervous system works like the animals, it helps us to define treatments. And there's lots of things that we can study in animals that we can't in humans that give us the information that we need. What we can learn from the rodent models, we could never learn in the human being.
Animal models are critically important for any research in this field to move forward. You can do modeling using other mechanisms and means, but in this instance, certainly the animal modeling has been critical to our progress, our discoveries, and now our ability to really change lives of people who are living with spinal cord injury today.
I think it's important to understand that when we test in laboratory animals, it's not just people who benefit from that. It is animals that benefit just as well. Our pet population gets many of the same diseases that people get. In the process, we're not only benefiting humans, but we're benefiting the animals themselves.
We really are working in a time of bench to bedside. I mean, everything we learn in the lab allows us to go back and forth between clinical trials in humans, the preclinical work in mice, at the bench, in test tubes, and they really inform each other, and they will allow what's called translation from bench to bedside.
Animal research is that critical piece that is going to allow us to go forward. There would be no new drugs and no new devices without that type of research.